Hello, everyone. I am Anna Bernasconi. I am a research fellow at Politecnico di Milano. I work in tight collaboration with Professor Stefano Ceri. Professor Stefano Ceri leads a big group of research in the fields of data management, databases, and bioinformatics. Currently, our group is composed by a full professor, an associate professor, 2 research fellows, including me, several PhD students, and master thesis students. Professor Ceri won an advanced ERC grant that lasted from 2016 until 2021. The title of the project was Data-Driven Genomic Computing, in short, GECO. During those 5 years, we produced several results that demonstrate how data-driven computing can effectively support human genomics research, in particular in the context of next-generation sequencing, NGS. In our project, we first produced a data model called the genomic data model for genomic regions that are captured by NGS experiments, and also a language called the genometric query language to express complex biological queries that are able to process big heterogeneous genomic datasets for tertiary analysis. Then we also supported federated database solutions for distributing genomic data processing, and we built a very large repository, now about 9 terabytes of data, that is called a metabase, that allows an integrated access to large data sources, such as the Cancer Genome Atlas, TCGA, or the Encyclopedia of DNA Elements, ENCODE, or 1000 Genome Project, or Roadmap Epigenomics. Our search engine was named GenoSurf, as we actually search or surf into the genome data. Finally, we also produce several web services for specific biological needs. During the last 2 years, we were prompted to test our knowledge and methods on an unprecedented scenario, the COVID-19 pandemic. Inspired by our work on genomic data integration after the outbreak of the COVID-19 pandemic, we turned our attention to the analysis and management of viral sequencing, aiming at reapplying the data-driven paradigm that we use for human genomics. First of all, we observed that the need for data integration across heterogeneous sources that collected viral sequences was real. So we decided to model a consensus conceptual schema for both metadata and sequence properties. For example, mutations of viral sequences resulting into changes of the amino acid residues that form proteins. And we thus built search systems over sequence repositories with also powerful data visualization tools. Remember that in the GECO project, we had already developed the GenoSurf search engine for datasets describing the human genome. So at the beginning of the pandemic, we noticed that there was no such system for viral sequences. We decided to implement such system, but to better understand its requirements, we interviewed about 20 expert virologists from all over the world. The result was a user-friendly system that we call VirusSurf. Any researcher can connect to it, perform queries, for instance, about when a viral mutation started and how it has spread in the world. Well, to find important genotype-phenotype correlations, well-defined phenotypes need to be ascertained in a quantitative and reproducible way. Also, the activity of sampling from clearly defined case and control groups is fundamental. For this reason, many efforts in the scientific community have been dedicated to harmonizing clinical records of COVID-19 patients. The Host Genetics Initiative was one of the very first large efforts in this direction. It included researchers and clinicians around all over the world, and thanks to the commendable commitment of its main leader, Andrea Ganna, the initiative was able to start very early during the pandemic, being able to build a concrete reference point for everyone that was studying the human genetics of the COVID-19. Andrea asked us to collaborate with the initiative, bringing our background on data modeling applied to life sciences cases. So at the time, we gladly engaged in the design, structuring, and harmonization of a comprehensive data dictionary that could help with the submission of individual level data. For this collection of requirements and the cooperative design of this dictionary, we decided to employ a Slack channel where we were able to coordinate about 50 clinicians for cooperatively designing the patient's clinical phenotype definition. At the end, the phenotype refers to severe patients who were hospitalized, and it has about 200 clinical variables that have been progressively consolidated and annotated. Well, challenges, many challenges. You should take into account that the dictionary was designed in a particularly early moment of the pandemic. Thus, the first challenge was related to uncertainty on the model content. Little was known about the implications of the disease and about which parameters could be measured to inform clinical and genome-wide association studies. This, of course, brought a great heterogeneity in the proposals of different researchers. Each one provided her or his own perspective, trying to tackle the problem in different ways, inspired by their intuition. So our goal was to provide a reconciliation model to include all reasonable perspectives in one shared model. Then probably the second most difficult challenge regarded the absence of agreement between different working groups on the information that should be gathered with priority. So for this, we chose to follow a comprehensive approach by not ever excluding fields unless they were redundant, organizing them in several categories that help understanding their broader scope. For example, type 1 diabetes is a comorbidity that regards the immune system, whereas asthma is a comorbidity of the respiratory system. Whereas the presence of dry cough is instead a symptom that is measured at admission. We then propose 2 levels of detail, fields necessary for longitudinal studies or for any other study. So, for example, the presence of dry cough could be, yes, measured once for all as a symptom, as admission, but could rather also be measured multiple times for a longitudinal study. As a third challenge, I would probably mention the difficulty in defining a clear and shared end user of such model. Was it the medical director who filled the health records of the patients at the hospital, just at admission or at every medical encounter? Or was it the person affected by COVID-19 who at the hospital, or most probably at his or her home before or after hospitalization, would keep record of information on her or his status on a dedicated smartphone application? Well, the purposes of this model were multiple and diverse. So we prepared it in such a way that all such cases could be captured. Finally, the biggest challenge was that of communicating the basic principles of data management to experts with a different background. We coordinated more than 50 clinicians who provided their own specifications of a good COVID-19 patient assessment. Of course, everyone had their reasons to suggest one solution or the other. So we addressed this complex scenario by providing a defined schema of attributes divided by category, each associated to a question in natural language and optional response values, requiring either a single or multiple choice. And each attribute was collected only at admission or during the course of hospitalization at several time points, either by a medical director, a person at the admission desk, or by the patient itself, following different input procedures, of course. Well, the benefits of establishing a unified model are several. Let me try to explain why. A unified model has the purpose of being accepted by everyone, so that several studies can all refer to it. Once studies are homogeneous in the collected data, it is then easier to make their dataset interoperate, meaning that they can be merged to answer broader research questions, or they can be compared to highlight differences in the observed populations. So the schema heterogeneity and consequently also the data heterogeneity are one of the first reasons for the inability to reach general conclusions in clinical and genome-wide association studies. Any means that favors the collection of homogeneous data will eventually support stronger research. Specifically, in the context of COVID-19, one direction that we encouraged with enthusiasm is the possibility to connect patient clinical information with the genetic aspects of both the virus and of the patient. To connect such three different kinds of information, the patient phenotype, the patient genetics, and the virus genome, it is highly important that the single databases are well crafted and designed to accommodate big amounts of data with a shared schema. A better linking among databases and correspondingly improved communication between specialists in the various disciplines will help us to understand better infectious diseases and to empower a richer precision medicine. Just as an example, in an early study of 2020 by Lee and others published on Cell, it has been studied that the co-occurrence of certain lab-generated mutations modifies the antigenicity of the SARS-CoV-2 virus, and therefore its sensitivity to specific neutralizing monoclonal antibodies. This kind of knowledge, when properly structured and applied in a hospital context dealing with COVID-19 patients, can practically inform treatment decisions of clinicians on given sets of patients whose clinical profile or infective viral strain is already known. Thank you very much for this interview and the possibility to speak about our contribution to the Host Genetics Initiative. Thank you.